Hello, everyone, and welcome back to the third part of the module. We're talking about engineering the excretory system. And so far, we've discussed the anatomy of the subsystems and the organs involved. Not all of them, but the most important ones. We've looked at the lungs, we've looked at the kidneys and liver, and we've looked at our skin. And we've also discussed replicating human organs as a whole in class, and we've discussed some of the challenges of that, some of the most recent advancements and approaches. So we've gone quite unconventional with this chapter, and now we're going to look at the continuation of those systems that are there to assist the excretory system. We're going to look at the specifics of materials and devices used in these applications. And then we're going to move on in the next part to diseases of these organs. So without any further ado, let's jump in and continue the talk that we had already. So as I mentioned, we're looking at these last 2 points here. The mechanics of the excretory system we will discuss later, but for now we're just talking about the biomedical devices, implants, and innovations. This was what we discussed in class last time, and it's the idea of oxygenation outside of the body or extracorporeal. And we went over a couple of different designs, and we realized that this was the most recent version, which includes an intertwined mesh of these hollow fibers that are very tiny, in the order of a few tens to hundreds of microns, and the blood is going to flow through this mesh as air and water pass through the fibers. And so the idea here is that the blood will have contact with air and also with the water flowing through, which will regulate its temperature and the oxygen concentration. Similarly, the CO2 concentration. So it's basically getting balanced back. And these flows have to be really accurately gauged and controlled so that you make sure that the blood exiting has the proper properties. Now, there's something important about this system, and we already talked about this, which is the resistance is quite high. As you reduce the size or the separation between these tubes, the resistance towards the blood flowing in between them is going to increase quite significantly. We'll talk about that a little bit later. The circuit that we showed is a simplification. It's just showing a pump, the oxygenator, as the blood goes back into the body. But the reality is that it's a bit more sophisticated, okay? The additional components that you see here, again, there are a few more in the real case, but include a heat exchanger. And the reason we have this heat exchanger is that sometimes the thermal exchange in the oxygenator region isn't sufficient, and you then have to heat it up again. Why do you need the pump? Because the pump is actually compensating for the pressure drop as it goes through the oxygenator and as it goes through this entire circuitry. And then you also have some injection sites for heparin and other fluids, as you can see in this image, which actually help with the anticoagulation, anti-adhesion properties of the oxygenator so that you can actually avoid thrombosis and platelet collection. Another system which is used in assistance to the excretory system organs, this time the kidneys, is the dialysis system. So it's kidney dialysis membranes. They basically have a very similar physics, a very similar fundamental working as to those of the ECMOs, the extracorporeal membrane oxygenators. The only difference here is we're looking at the exchange of fluids and molecules, and you have a dialysate, and there's usually an exchange between these two through the membrane, which is very similar to what you have in oxygenators. But in oxygenators, you've just got the oxygen molecules coming from air or the pressurized oxygen through the pores of the membrane. Here you've got a liquid, a fluid on the other side, which actually is having diffusion from it entering the other side. So similarly, this is usually utilized when you actually have a lack of performance in the kidney. And what it does is it basically maintains the balance of the body by getting rid of all the waste products that the kidney used to get rid of, and basically regulating those levels of biochemical species in the body. And there are two approaches, of course. One is to utilize a process that's inside the body. It could be a catheter, and blood is basically using this catheter, which is a tubing inside the body, and it basically fills the body with a dialysate and then removes the dialysate afterwards. Or you've actually got an artificial kidney, which is basically that entire membrane system connected to your vascular network. So in either case, the situation is that you have to have a membrane, and this is the typical case where you're drawing blood from the arm, and then you're feeding it through the dialysis membrane, the dialysate, which is actually going to exchange the molecules with the blood, is also being put into this membrane chamber. And as the molecules diffuse into the dialysate, which is now used, has to go back in the container here, and the fresh dialysate comes back and diffuses more and more of the blood into it. And then you actually have the blood that's now being cleaned of all of the toxins and waste back into the body. And these membranes really, on one side they have blood, on the other side they have a fluid, which is the dialysate. And this dialysate, you have to basically constantly refresh its content because it is getting all those waste molecules from the blood into it and storing it there. So this is the typical, which is what you have in hemodialysis. And then, peritoneal dialysis, which I explained earlier, you've got a catheter, and this catheter is where the body, basically the dialysate inside the body fills up in it, and then it actually removes later on with the proper removal towards the bladder or other organ of exchange. And it's interesting to know that actually there's a huge population of patients that are on dialysis, kidney dialysis systems. It's about 1 million worldwide, which have to get 3 times every week. It's pretty huge process, and it takes a huge toll on the patient as well. So really is also as challenging, maybe not as challenging, but quite challenging. It's comparable with the membrane oxygenator systems. The system used to be very similarly to the oxygenators, a single membrane, and it actually, or you stack those membranes on top of each other, and you ran the blood through these layers, these 2D sheets. And then there was this was the flat plate, and then you had the twin coil, which was a bit more complex. And in this case, as you can see in this image, you've got 2 chambers here, 2 pockets, which are super long, and they get wrapped around this core, just like, toilet paper, for instance. And then blood goes through and comes out. But the idea is that blood comes through one channel, dialysate through the other channel, and they can exchange through this wall in between them, which is made out of this fiber material behind. And so as they travel around the fiber through that cavity and come out from the other side, they get time to exchange the waste products. So the reason with this is that it's a lot more compact. Okay, so you're utilizing that same surface area across a very smaller volumetric footprint. And then again, most recently, hollow fibers have come to the aid of these systems, and we've made lots of innovations on that front. And you can see numbers that are quite staggering. You've got 11,000 fibers per a very small tube. That means the fibers have to be in the order of 200 microns in diameter, and super long, really, you know. So the ratio, the aspect ratio is super huge, because they're about, you know, 150 millimeters long, and then just a quarter of a millimeter wide. So they're very easy to damage and break. And what any of these materials need to have is a proper contact performance with blood and the dialsate. Okay, they need to wet blood so that it can actually pass through, and they also need to wet dialsate because this is designed for these two components. And the material is usually cellophane. It could also be polyethylene glycol, polyethylene terephthalate, and those copolymers are more biocompatible, and but they also provide a good level of filtering property. And this filter size here is 2.5 microns. So the pores in the membrane are actually 2.5 micron. Now, here is what the most modern kidney dialysis process looks like. So it's not a membrane. This is the hollow tube. That's a mistake there in writing. But the problem with these, as you can guess, is because these tubes are very, very fine, the flow of blood through them can face many challenges, including stagnation. It basically, in some regions, what it does is you have dead flow. Because these fibers are not perfect, because the pores are not perfect, they form a rough surface for the blood to flow over. In some regions, as the blood enters, even in the entry region here, not even through the tubes, you can have stagnation, you can have areas and regions of zero flow or dead flow. And then the other problem is turbulence as they travel through, right? And as they go through various sections, including the dividing section there, the coagulation, collection site here on the other end, you can see turbulence. And here is where you actually, in these cases, you put the dialsate there, and it runs again around these, basically over and around these fibers. And that's how the exchange happens through the walls of these fibers. So the efficiency, because of that turbulence and dead space, can be sacrificed. But apart from that, you have huge amount of resistance there, which actually requires a pumping mechanism to compensate for. And the other thing which is really important is that the clinicians should actually degas or de-air the chamber, prime it initially before you start, which makes it very challenging because you now have to deal with a much more tricky problem of having an initial priming volume that fills that up, and then you continue the process. You need to draw blood from the patient in order to be able to do that. And if you have a little bit of air there, those bubbles, those air gas bubbles can go into the bloodstream, and that's really not something we want, okay, because they can cause various cardiovascular issues. So the other challenge here, or it could be a challenge, maybe it's a good thing, is the viscoelastic property of blood, meaning that the viscosity of blood changes as you change shear stress or shear rate. And the reason for this is that blood isn't just a liquid, it's a viscoelastic material. It has both viscous properties and elastic. And depending on what shear stress or shear rate you actually put on it, it can experience different amounts of elastic and storage modulus, or elasticity and viscosity. And this graph here shows you what that's all about. The viscosity actually is dropping as you go forward. And is that a good thing or a bad thing? It's actually a very good thing. It's a very good thing because you're making blood less and less viscous and easier for it to flow through the channels here. Okay, so as the viscosity drops, it becomes easier to flow through those tiny thin channels. That's how this shear thinning effect, the reduction in viscosity, can help us. And these have been around for a very long time. Now we're going to talk about their materials. They've been using cellulose very widely, but nowadays they're not using it anymore. The reason for that is that they're actually not as biocompatible, and their performance is also weaker. So they've moved towards synthetic versions of these membranes, and those include polysulfone derivatives, polyacrylonitrile, PANS, basically, that's the acronym we use. Polymethyl methacrylate, PMMA, another polymer that we commonly use, and it's a light activated one as well. It's got basically you can cross-link it with light, and then ethyl vinyl acetate, which is EVAL, and these all are synthetic equivalents of cellulose, which you can conjugate and use in combination of each other. But the main thing is they've got much better biocompatibility and performance than those. But still, a big issue is whether or not they are hydrophobic. Okay, because if they are hydrophobic, that's a big challenge with platelet adhesion. So that's why they actually use additives in order to improve their biocompatibility or their lack of absorption of the molecules. And if you now look at the fibers in more detail, they have various designs. So depending on each of these are a big name brand, Brown, Baxter, they're all very huge, 3M. And what they specialize in is a specific chemistry, okay? So for instance, here 3M's membrane has polyether sulfone, okay? And you can see that depending on what material you choose, the pore size is going to be different. So if the pore size is different, that means the diffusion efficiency, how well the material can get through, is going to change. So that's why selecting these materials. And how do you induce this? Well, the chemical process of manufacturing that under pressure, and then whatever that is, if it's, spinning it, whatever, you actually get various sorts of composition. Now, this is cool because if you zoom into each of those pores, they very much have a tubular structure, which is actually the path through which blood should make its way through. So when we look at this law that we had a while ago, now we've used it many, many times, resistance has proportionality with the inverse of the fourth power of radius. So if you increase radius, resistance goes down significantly. If you decrease radius and decide to, considerably make these pores narrower, that means that to the fourth power, this resistance is going to increase super strongly, and that means that you will actually have less of the exchange of materials, and that's just not what we want. Another area is the diameter itself, right? The diameter itself is also governed by this equation. So the flux going through this, if you reduce that by a factor of 2, the resistance here is going to change by a factor of 16. okay? And that's when the flow rate is going to change, and our chances to need less pressure going to reduce. So you're actually going to need more and more pressure as you actually make these narrower. So the same dynamics happens here. You have to decide what you want. But in any case, these pore sizes are not a single fixed pore size. They actually have a distribution. And they go all the way from very tiny pores to larger ones, and we'll show you experimentally. But the idea is that this distribution here has peak. And that's really the average pore size, the most common pore size that we have amongst these. These three graphs are pretty interesting. I'll start from this side. It tells us that as you increase the transmembrane pressure, okay, you're increasing ultrafiltration. Or in other words, if you increase ultrafiltration and it filters more and more, that means the pores are finer and finer, then the transmembrane pressure goes up because there's going to be finer holes to travel through, means that you need more pressure to do that. Here you've got blood flow versus clearance. I'm not 100% sure about this, but similarly, I think it's the gap between these holes that we're referring to here. And as you increase it, you will get a greater blood flow for the same pressure. Depending on what structure you're talking about, this can vary. And this is another interesting one. It's the filter performance. What it's saying here is it's not filtering at a certain molecular weight sharp. It has a range of things. So it can do a little bit more than that molecular weight, a little bit less. But ideally, you want to have a molecular weight that you're interested in safely positioned so that you only get the proper molecules and substances to pass through, and not everything. Finally, you can see that depending on what the pore size is, you can have various performances. So in this case, you're looking at using this for convective therapy, meaning that you've got convection flow, bulk flow, traveling of liquid through the pores. In this case, the pores are so tiny, you have mostly diffusive therapy, but you've also put convection there, meaning that you think molecules will diffuse, but we won't have a motion of liquid through this. And here you've got a case where you've got only diffusion, so it's super thick or whatnot. So the other aspects of these are, of course, they're all made out of synthetic polymer, apart from this one, natural polymer. Hydrophilicity, we want them to be hydrophobic to some extent, so that you don't have platelet adhesion. So there's a variety of things you have to look at in these. And then you also want to basically consider the diameter of these, the number of these, how much flow you're going to have. And here you can see the hydrophilic versus hydrophobic, a good graphic that shows basically how water is going to travel through these pores. And you can see that if it's hydrophilic, it's going to fill in the pores and travel easily. If it's hydrophobic, it's going to make its way and make these tiny droplets and never going to meet the other end. So that's also important about the hydrophobicity. So we're going to conclude it right there for this session, and then we'll come back, and in the next one, we're going to actually talk about skin. So for now, take a quick break and we'll be back.